So, good afternoon, everyone. Many thanks for joining this very special seminar. Today, we have a very special speaker, Professor Yong-Guan Zhu. Actually, Yong-Guan did his PhD here roughly 26 years ago, and from 95 to 98, right? Yeah. And at that time, he worked on the plants and on one of the radioactive waste from the nuclear reaction is a cesium, right? How to, yeah, to see how that radioactive chemical transfer from the soil system to the plants. And later on, Professor Zhu worked in Australia and then moved back to China. And his famous work is on the rice system because our metal contamination is a big issue in China and many other surrounding Asia countries. And his work shows how the arsenic transfer from the soil system to the plants and the risk to the human population. And now I think Professor Zhu's work more focused on the microbiome. And these are the exciting things that Professor Zhu is going to talk. And Professor Zhu is a very influential scientist worldwide, and because published many papers, over 300, I think, in international well-known journals, and also many papers have been cited over thousands of times. And he got a very famous award from the International Union of Soil Society as one liberty, liberty, and only one scientist can get that award every 4 years, I think. So, so this time, Professor Zhu wanted to come here, establish new collaboration, and also wanted to show this side lots of good memory, right? Yeah. So please join me. Welcome, Professor Zhu. Thank.
Okay, thank you so much, Dr. Liu, and also Professor Bell for your invitation. And it's great to be back in Field Park. I haven't been back since I left '98 in June, and I try to establish some more collaborative link with Field Park. So this is such a wonderful and influential center for environment and ecology, evolution biology. And I think we share many common interests in microbial ecology. And today I will particularly talk about antimicrobial resistance as one of the issues we are facing globally right now. But to start with, I want to just say a few words about where I come from. I'm from the Chinese Academy of Sciences, which is the largest research and development center of China. We have over 70 years now, 75 now, we are celebrating this year. And it is the key center for national science and technology innovation. And we have so-called 4 arms. Super University. We have a couple of universities. The largest is called the University of Chinese Academy of Sciences. We offer high degree mostly. So about 60,000 graduate students we have, because that's distributed over 104 research institutes. And it is a think tank and also an engine to drive national, we call this is a innovation-driven economy right now. So the Chinese Academy is really the major engine to drive that. And we are also a pioneer in developing new management systems in education, in science, and technology, because we have so many graduate students, and we also have young talents program for the future, for the next generation. So this just illustrates how it's scattered around the whole country. And I'm currently based in Beijing, but I used to be based in Xiamen. We set up a new institute there about 20 years ago, Institute of Urban Environment. And the institute I'm in right now is to be 50 years anniversary next year. It was set up right after the International Human Environment Congress in Stockholm. It's the same time as UNEP was set up. So this is the whole picture of the size of our academy. So we have 2 universities and many state key laboratory, and the staff scientists is nearly 70,000 staff scientists, and we have cast members about this over 700, and we also have British foreign members. They are part of our academy, and our current budget is around 50 billion Chinese yuan. So now I want to move to the science part of my talk. As we are all very well aware of this globalization of this planet, and in particular is the microbial world. It's increasingly being globalized because microbes move across boundaries without passports. When I travel with my microbiome. When you import commodities, you import microbiome. And the migratory birds, they can also transmit microbiome. So microbial movement at the global level is becoming more integrated and more intensive, I would say, due to globalization of the economic system or the globalization of the human society. So we often say that one Earth, one health. So I just mentioned this global trading and the wildlife migration, and also at a local scale, these microbes can move from soil to the plant, to the insects, and to people. And there is an intensive microbial cycling within the local ecosystem, but also transport globally by these processes I have just mentioned. So, and all these processes are controlled or driven by human activities, like the discharge of wastewater, and also planetary actions like climate change. Warming will expand the range of the animals, and they carry this microbe and also expand in the same time. So we also say this is an issue of planetary health because they are indeed driven by planetary actions. And over the last 20 years, alongside the topic of arsenic in soil plant system, I also developed this program at the earlier stage. 20 years ago, not many people are really interested in antimicrobial resistance. And we started to look at first antibiotics as a chemical residue, but we realized that the real impact is from the antimicrobial resistance. The chemical residue is not of big importance to human and wildlife health, but the induction of the resistance is the problem for global health system. So we, at the beginning, we also take the whole ecosystem as our research framework, because each component of the ecosystem is all tightly connected. So they are not in isolation. So we try to understand the dynamics of the microbial cycling within the entire ecosystem. Of course, my background is soil science, so we focus a lot on the soil, but soil is not in isolation. So soil plant system, soil movement from through water erosion to the rivers, etc., and also to groundwater. So we are trying to understand the interconnectivity of antimicrobial resistance. And talking about antibiotic resistance is a pure incidence. We know that Fleming and Chen, they discovered the penicillin. And also Florey, actually, he was a graduate of Adelaide University, where I did my postdoc. So my research really has some intellectual roots with these 2 organizations. And also this Professor Waxman, who discovered the streptomycin, he actually is one of the pioneers. I would say he is the father of soil microbiology. He defined the discipline. And he and his team, discover this streptomycin actually from the soil microbiome. So now we know that the soil is actually is a repertoire of many different drugs is waiting for us to explore. So the metabolites from bacteria, fungi, they can be utilized as a pharmaceutical compounds, like and even anti-cancer compounds can be discovered from the soil. And because of this, the impact of antimicrobial resistance onto human health, both United Nations and this nature also put a special issue on it. And now actually not only WHO, but FAO and the UNEP and the Organization of World Organization of Animal Health, these 4 organizations are united to work together to understand the dynamics of antimicrobial resistance. So that is why we consider this issue should be under the framework of One Health, because animals, plants, and human, we share the same microbial community. And many of the pathogens, they can be infecting different species in the same time. We call it zoonotic pathogens in the environment. And through the literature, we can see that not long after the Nobel Prizes in antibiotics, this paper in Nature conducted by DEFRA in England demonstrated that the use of antibiotics in pig industry is beneficial because antibiotics can promote the pig growth, and it's good for the industry. However, only after 20 years, there is another study, also in Nature, demonstrates that some of these antibiotic-resistant plasmids, they can be spread from chicken to chicken and also from chicken to man. So this is probably one of the earliest studies demonstrate the One Health concept of microbial movement in the environment. So this microbial infection is not just happening in the hospitals related to our health, but in the general environment. It's already it's influencing our health. So now, at the moment, this I think is most recent study demonstrated that globally, it was estimated that about 5 million people every year, 5 million people death every year is related to antimicrobial resistance, and about 1.3 million people is directly attributable to bacterial AMR. So you can see the scale of the problem. And the problem, I would say, it's deeply rooted in the ecosystem, because in the ecosystem, there is such a dynamic microbial movement. So we consider antimicrobial resistance, and the determinants, the genetic determinants are we call it the antibiotic resistant genes. They are considered as emerging contaminants. Because, for example, we are working on heavy metals. Those are traditional contaminants. They cannot be amplified themselves. But the antibiotic resistant genes, they are carried by live bacteria. They are a piece of DNA information. So they can be amplified exclusively if the conditions are right. So even one single bacteria, if it carries resistant genes, and they can amplify the genes very quickly. And as I have said, that they can be transported globally when we move around, or the commodities move around. They can be transported in longer distance. But more importantly, they can be sprayed horizontally. Under chemical stresses, these bacteria, they can exchange genetic material, so-called horizontal gene transfer. They can be stimulated by the residues of antibiotics, but not just antibiotics. Many other chemicals in the environment, they can stimulate the exchange of these resistant genes. And these resistant genes often, they're clustered together on so-called genomic islands. Many resistant genes, they're clustered together. So in the past 20 years, we have been focusing primarily on two major sources. One is the urban wastewater treatment plants, obviously, that collect our gut microbiome. And the other source is intensive animal farming. So that's we use so much antibiotics in the past. Although now globally try to phase out, but still you use it as therapeutically, and some maybe still use occasionally as a growth promoter. So these two pathways that discharge this antibiotic resistance into the environment, and they can come back into the human living environment that cause health issues. So this is just one of the first studies we conducted a systematic evaluation of the resistance contamination of the environment. So we collect fresh manure from this, for example, this pig, this particular farm, we collected this still warm. So we get this fresh manure, but also we collected the organic fertilizer, it's a manure compost. And we also collect the samples that the soil has been fertilized with organic manure. So we take three major big intensive pig farms, and we use high throughput quantitative PCR. Use this PCR, we can quantify over 300 functional genes in the same time, together with 16S. So we can calculate the copy numbers in each sample. And you can see from this heat map, for the fresh manure, it can contain up to 10,000 times higher than the background manure. That is the manure from a pig. It's not fed with antibiotics. So this resistance is highly elevated. And using this machine, we can quantify so many diverse genes. And we find that over 100, sometimes 200 genes can be detected in the same time from this pig manure and the soil samples. Sorry, this is Chinese. I didn't realize. Yeah, so basically this diagram actually tells you in English. So you can see that this is the control soil, this is the manured soil, and this is the compost, and this is the fresh manure. So this can separate it very nicely, and the separation is driven by, you can see like quinolone, also by zinc, copper, arsenic, and tetracycline. So this diagram actually tells us that it's not just that the antibiotics are driving the separation of the resistant to all in those samples, but also these other chemicals that can induce chemical stresses that can also spread the resistant genes. And I'll come back a little bit on that again. And we found that these resistant genes correlated very well with the transposase. So one of these mobile genetic elements we measured from this sample. So that again shows this horizontal gene transfer is happening in those environmental samples. And we have more data to demonstrate that. So this is one major source is live intensive animal farming. And another source is from wastewater treatment plants. We sampled major cities in China, across China, 17 cities with 32 wastewater treatment plants. And I sent my students to those different cities and asked them to do that in the early morning. We try to catch again the fresh excrements from humans in the morning. And in this case, we use the metagenomic sequencing, and we calculated that each Chinese person, every day we discharge that much copies of resistant genes into the wastewater treatment plant. This number, I calculated that if each copy is 1 second, that number of copies equal to 3 million years. So you can see how big is the number we are discharging to the wastewater treatment plants. And what happened to these resistant genes? So using this metagenomic, we found that there is a core resistome. Despite the wild geographical distribution, we found that over 95% of the resistant genes are shared in those wastewater treatment plants, despite their geographical distance. So this clearly shows that the resistance from those systems are driven by human activity, not the environmental conditions. And we also try to look at the dynamics. So if you look at this antibiotic resistant genes copy number per bacterial cell, there is a good reduction as compared to the influent. This is in the sludge, and this is from the effluent. So we discharge the wastewater after treatment. Chemically, it's safe, it's clear, but it still contains resistant genes. Over 2 copies per bacterial cell. This is compared to probably 0.01 or even less in the natural environment. So very little copy per bacterial cell. So you can see that we still discharge substantial copy numbers of these resistant genes into the general environment. And that is something we must understand the potential risk to humans. So with that, we propose, in addition to intensive animal farming, we propose that wastewater treatment plants are potential hotspots of superbug, potential discharge of those superbugs into the environment. But we still know so little about their fate in the environment. We don't know their fate in the environment. So for that, we conducted a large-scale national continental scale survey. We took samples from 18 major estuaries in China. We take the estuarine sediments. So the sediments is like the archive of what we discharge from the entire river system. So from here, you can say we detect on average over 115 genes discharged. We can detect it using high throughput quantitative PCR. And on average, we detected 0.66 resistant genes per cell, which is lower than what we discharged. Of course, when we discharge, there is a dissipation process, but still we see there is nice evidence showing the spread of the resistant bacteria into the environment. And this is what I call as a microbial footprint of our human society. We have water footprint, we have carbon footprint, and this is the microbial footprint is recorded in the estuarine sediments. And there is a potential risk into the human living environment. And even given this, all the samples actually we get from the natural environment, we still found a very nice correlation between the absolute abundance of ARGs with the level of integral is another genetic determinant for the horizontal gene transfer. So again, for this large scale study, we demonstrate that the mobilization of these resistant genes in the environment is happening. It is probably happening. And then we even have more data showing that those integral cassettes we cloned from the environment, they contain diverse resistant genes. So this indeed showing that these resistant genes in the environmental microbial community, they are potentially transferable and they can find their way into human pathogen to cause the emergence of superbugs that will cause therapeutic failure in the hospitals. Now, as we all know that the natural biome follow the principle of distance decay. So this is also true for microbiome. With increasing geographical distance, the similarity decrease substantially. However, for the resist storm, we don't see such trend. So the environmental conditions are not affecting the composition of the resist storm we observed in the environment. Then what are the driving forces? We use the data we measured from these samples together with anthropogenic factors like the population, the volume of wastewater discharge, and the volume of animal industry. All these factors combined together contributed over 60% of the variation we observed in the resist storm. So you can clearly see that this human activity is driving the spread and the emergence of this resistance in the environment. And we already showed that the mobile genetic elements are contributing to this spread process. And from the river or from the wastewater treatment plants, in many cities, we are trying promoting so-called green city, try to reuse the reclaimed water. So in this study, we sampled many urban park soil. Those urban parks have been irrigated with reclaimed water. So we found unanimously with the long-term irrigation of reclaimed water, there is an accumulation up to 1,000-fold increase as compared to the soil in the same city, but in a pristine environment. So you can clearly see in many of these cities, if the urban park soil has been irrigated for a few years with reclaimed water, there is an accumulation of these resistant genes. We don't see the immediate risk, however, because we don't know yet how to quantitatively assess the microbial risk associated with antimicrobial resistance. But we do see very clearly there is an elevation with long-term irrigation with reclaimed water. So we took this study further as an ecosystem view of antimicrobial resistance. And now we consider this microbial cycling as our key research topic. So previously we studied a lot on biogeochemical cycling of nutrients, contaminants. But I think along with this biogeochemical processing, microbial cycling is something we still know so little. And we must have developed models even in the future to predict the fate and to predict the risk. But more importantly, probably we need more parameters about the horizontal gene transfer, the frequency of horizontal gene transfer, and the drivers, the chemicals. There are millions of chemicals are now discharging every day. And what the impact of chemical mixtures on their spread of these resistant genes in the environment. We know so little. So for some of our study in the past, we look at the soil plant system. We even can get the xylem sap from the plant, and we can detect the resistant bacteria within the plant. So you can see the microbe can move within the plant and also on the surface of the plant. So this is something as one of the topics for ecosystem studies. And we also look at the long distance transport of the resistance or the resistant bacteria in the environment. And one of the topics we are doing at the moment is really on the biofilms that colonize on the plastic surface. We call the plastisphere. So you can see the plastic surface at the beginning, and after 30 days, you can see this entirely colonized by bacteria, by protists, by viruses. So we found that there are many viruses actually also carry resistant genes. And this just to show you the enrichment of in comparison to the surrounding water, we can see that on the plastic surface, there is relatively more pathogens. And we know the pathogens has the capacity to adhere on surfaces. So we believe that those pathogenic bacteria probably have some advantage in colonizing the plastic surface. And after they form the biofilm, actually, we found that they are becoming more resistant to antibiotics. You can see that we use Raman spec together with heavy water labeling. So if we can detect the intake of heavy water using Raman spec, that means those bacteria are live. And you can see with increasing concentrations of antibiotics on the plastisphere, we don't see any decline in activity of the bacteria. So they stay resistant or tolerant, at least. To be safely, we say tolerant. They can survive under antibiotic additions. However, in the free living, in the surrounding water samples, we see very nice decline with increasing antibiotics. So from this study, we can clearly show that they are colonizing the plastic surface, they become more tolerant. And with time, probably they develop resistance in the end. And they can be transported together with the plastic in the river system into the ocean. So this is just a laboratory study. Then we go further into a river. We take a small river, over 100 kilometers small river, to study the whole river process. We put the plastic samples in different parts of the river. Some of the samples, we put them in the waterhead, in the pristine environment. Indeed, you can see those samples contain very little resistant genes compared to this peri-urban and then in the urban system. This is antibiotic resistant genes. These are mobile genetic elements. They show the same trend. So we can clearly see that with more population, with more human activities in the live river system, we demonstrate that the resistant genes can be elevated by human activities. So most of these studies in the past, we have focused on DNA. We talked about many of these bacteria cells, probably they are dormant, they are doing nothing, or some of them may be dead, or some even is from extracellular. So at the moment now, we are spending a lot of efforts in trying to understand the active microbial community from those environmental samples. So in this sense, we particularly use this heavy water labeling, and we use the Raman spec. With Raman spec and with the sorting equipment, now we can pick up each single bacteria cell. We can either do single cell sequencing, or we can do a mini metagenome. And from that, we can actually find many uncultured bacteria. Probably we can understand their identity and their function. So for example, from this study, we compare the soil from Tibetan, a farmland soil, a manured farmland soil, use this heavy water labeling. We can clearly see the difference. This normalized carbon deuterium ratio, that means it's active, the proportion of active bacteria in the sample. You can see much lower in the Tibetan pristine soil, and in the agriculture soil is much elevated. So indeed, using this new technology, we can pick up the active bacteria cell from the environment, and also we can do the sequencing. So from each bacteria cell, we can get through sorting, and we can link this phenotype to genotype. And luckily, we got one we identify one uncultured potential high-risk pathogen. This is a pathogen that cause, particularly for young women, you have a skin rash or something. This cause acne. So this particular Cutibacterium, we through this metagenome, we can bin the whole genome, and we can get this resistant genes and also some of these mobile genetic elements and also the virulence factors. So from this single bacteria, single cell technology, actually we can sort out these bacteria cells and also to link the phenotype to the genotype and also to get the genetic information. But unfortunately, at this moment, it's still not high throughput. We are hoping to do it in a more high throughput fashion so that we can do perhaps millions of cells in the same time. And that is one of my purposes. Tomorrow we will have a meeting in Cranfield. We are talking about developing new microfluidic system so that we can sort the cells more efficiently and to do further studies. Now I want to try to quickly perhaps move to some of these final slides about risk assessment. So I mentioned that the potential risks actually is linked to the host of these genes, whether they are pathogens or not, or whether these genes are transferable. If they are already on the mobile genetic elements, that means they are more risky. If they are already in the pathogens, they are even higher risky, highly riskier than those in the environmental bacteria. So this is just one study. We haven't really published it. We again sampled, instead of the influence, we sampled the effluents across China. And we do metagenome analysis. And from this diagram, you can see over 40% of these resistant genes are carried by plasmid. So we can be pretty sure that what we are discharging from these wastewater treatment plants, a large proportion of these genes can be transferable. That is why we found this nice correlation between the level of integron and the level of resistant genes, given that all the samples are from real environment. So from this study, it is very clear that this horizontal gene transfer, it is potentially happening in the environment. And we now need more studies to get the parameters so that in the end, we can model that, we can predict that. So we also, you can see, we isolated some of these strands. For example, these two, particularly they are pathogens. And you can see they have these resistant genes. They have these mobile genetic elements. They contain these virulence factors. And some of them even contain heavy metal resistant genes. So those are the resistant determinants. They tend to cluster together, as I said, because those are the elements decide their adaptability of this bacterial cell. So those bacterial cells, they harbor the genes, they have more skills to adapt to stresses in the events of environmental disturbance. So it makes sense that those potential pathogens that do contain or harbor this resistant gene. So the hypothesis we had is that wastewater treatment plants are potential hotspots for discharge. And indeed, from this study, we can see it is quite possible. And now we try to look at their fate. If we discharge the water into different environments, then we follow the fate of these pathogens in the environment to understand their potential movement and survival under different environmental conditions. So I alluded a little bit on the chemical mixtures. I believe, Guy, you have been working on chemicals or the food web. Yeah. So we actually, in my lab, we do chemical mixtures nowadays, particularly like a pesticides, fungicide, and pharmaceuticals. So these are the chemical mixtures. They can induce resistance through co-selection, promoting horizontal gene transfer, and also shifting microbial community. So through these three pathways, the chemical mixture can also contribute to the spread of resistant genes. And this is something I call that the confrontation between the chemical world and the microbial world. And that is the world that we humans share the same space, and we must understand that. So just to illustrate, we did an experiment. This carbamazepine mazepine is a typical pharmaceutical compound to treat epilepsy. Although it's not so common, still it's the drug can be detected in most of the environment, in water. Even at the small level, we found that they can promote resistance, particularly when they are coexistent with antibiotics. They can drive further the spread of resistant genes. But for the sake of time, I won't go into details, but that is one of the topics we are now trying to do, to understand the impact of chemical mixtures on this disturbance of the microbial community. So with that, I want to wrap up my presentation today. I hope I have been I have convinced you that microbial cycling under One Health framework is an emerging public health issue, and we must take a holistic view of the environmental dimension of antimicrobial resistance, because we should consider both the food, the water, the air, and also the landscape, and also the long distance mobility, even the global mobility of these resistant genes. They can be carried by either commodities or birds or humans or by wastewater. And indeed, we need a paradigm shift in our research efforts. We must also, along with DNA-based, we must also move to culture-based, particularly to characterize the resistant bacteria pathogens. And with that, we must have new technologies to combine new technologies to understand the dynamics. And also, at the moment, we are more or less focusing on the status of pollution, but in the future, more on the quantitative risk assessment. And in the same time, we must understand the potential impact of climate change, warming, flooding. This will all drive the spread of this resistant gene. In the event of flooding, we often see the breakage of pipes of wastewater, and they leak the human pathogens into the environment. I think indeed these are the issues we can maybe collaborate to do some joint research. And I have been back in China for 22 years, and over the years I have been very lucky. I get funding from the Chinese Academy of Sciences, Natural Science Foundation of China, and also Ministry of Science and Technology. And I have many talented young students and postdocs. And I have wonderful collaborators, both from home and abroad. I have many collaborators in the United Kingdom and also in many parts of Europe. And with that, I want to thank you for your attention. And I hope we will have opportunities to collaborate and to learn from each other. Thank you so much. So I welcome comments or questions from the audience.
If we use the mic so people online can hear.
Thanks. That was a fantastic talk. I've got a question about as we're going through this stage now, kind of starting to quantify the risks and hazards, etc. I mean, ultimately, we're going to want to put in mitigation strategies or bioremediation, etc. Is there a sort of have you got any thoughts on what bioremediation might look like to try and minimize those risks?
Yeah. So I think that some bacteria, we call it bacteriophage, probably can play a role for those engineering systems. For example, composting system, and the wastewater treatment plant system. If we know enough knowledge about their relationship between virus and the bacteria, probably we can develop a cocktail remediation strategy for reducing the risk. But also we also tested that for some cases use a hyperthermophilic composting. We probably can reduce the resistance more dramatically. Normal composting cannot reduce the level, but a high temperature can. But in addition to bioremediation, we are also practicing like a chemical, like a biochar. If we turn the organic manure into biochar, we can pretty much eliminate the pathogens, and also in a way probably also degrade the chemicals and lock up the metals as well. So some chemicals and the biological, I think these are potential methods. Thank you.
Great talk. Thanks a lot. You mentioned just in passing about survival, and I suppose for all these resistance genes, once they're no longer being selected for by the antibiotics, then we presume if there's some kind of trade-offs that they'll be on the road to extinction. So what's the current thinking on what like the half-life of resistant genes might be?
No, there is some fitness cost to carry this resistant gene, but it is very difficult because there is already a background level. These antibiotic resistant also, they are the arm race in the natural community. So it would be very important for modeling purpose. We must know the half-life and the driving forces. Thank you. Maybe it is something we can collaborate.
Yeah, I think it's a really difficult question, but if you're thinking about parameters to measure, then it's one of these like source sink communities. You have all of this coming out of the sewage treatment plants, and then we sort of assume that they're dying off, and really the important parameter is how quickly they're dying. Thanks.
Philip Taylor online has a question. Philip, would you like to just unmute yourself and ask?
Yeah, thank you very much. Thank you very much for the talk.
I'm so sorry. We're having a bit of a hard time hearing you on this end. Let me turn up the volume. Try again.
Thank you very much for the talk. It's fascinating. My question relates to the use of antibiotics on crop plants. In some countries, a lot of antibiotics are put onto crops. And is that something that you've considered, or do you think that's an important aspect of your work?
Yeah, thank you very much. It is an issue. I think it's more of the issue is the fungicide, actually. So for the common antibiotics for bacteria infection, usually there is little use in the plant, but the fungicide is so commonly used, and the fungi resistance, antibiotic resistance is an emerging issue. So we have just started some small project looking at that. But I think fungicide probably is more for the plant and linked to human, I think.
Right. Thank you.
Thank you.
Thank you. Anybody else in the room or online?
Just elaborating on that. So do you mean fungicides from what perspective that plant? Yeah, like can you elaborate on why it's fungicides in plants?
Yeah, so example, at the moment we isolate the Candida. Candida is a kind of ear infection, I think. So the I don't know how to pronounce that the fungicide, something OZLE type of compounds. Azo, yeah. So they are similar between animal, plants, and humans. So those are the, when we use the common drug, that's a problem. I think the active compound, the mode of action is similar.
Right. Thanks. Can I ask another one? So I've seen this a few times in the literature. I don't follow it that much, but the correlation between heavy metal tolerance and antibiotic resistance, is that just associations that have developed during the recent industrial period, or are those historical evolutionary associations?
It is within the entire Earth's evolution system. Because, for example, mercury-resistant genes and arsenic. I have been working on arsenic, and some of these compounds, we believe they are primordial antibiotics, like methylated arsenic,
right?
Reduced form of methylated arsenic. They are very toxic to their friends or their enemies of the bacteria. So we can also find resistant determinants to methylated, reduced form of arsenic. So this actually is a entire Earth life evolution system. I think it's yeah.
Interesting.
Yeah, long billions of years evolution.
Cool. Yeah. Thank you.
All right, if we have no further questions, I want to thank you all for coming. Thank you so much, Professor Zhu, for coming and speaking and answering these questions. For the junior scientists in the room, that's masters, PhDs, and anyone around that level, postdocs included, if you would like to wait for about we'll do about a half an hour session of little chats in a very informal setting with Professor Zhu. We're going to move out to the couches just outside, just to keep it very informal. Yeah, if we have any questions, anyone has anything to ask, now's the time. Doesn't necessarily have to be directly about the antimicrobial resistance. All right, thank you.
Thank you.
Thank you.